Okay. So we'll get started today with sort of the last few slides from last week that we didn't make it to, and then we'll move on to all the slides from this week. So the last aspect that we covered last week, which actually fits quite well into this, is sort of the steps beyond just the initial segmentation and object analysis, where you actually start to look at things like the surfaces and the interfaces. And so with a lot of the things that we've done up until now, they've been really focusing on the bulk of the object. And so that means the volume, all of the pixels together. And we looked at things, like the shape tensor, where we're analyzing all of the shape inside of it as an ellipse. And now we have sort of the desire to add tools to look at the surfaces and the interfaces that we have. And so with segmentation, we've actually already identified the areas that are interesting for us, so the pixels or the voxels in 3D. And the interfaces are sort of one dimension lower from this. And so if we are looking at a 3D image, then these interfaces correspond to surface area. And if we're looking at a 2D image, this corresponds then to perimeter. And so the interfaces are very important in sort of the scientific fields outside of just the image analysis itself. And so if you look at physical, chemical, and biological processes, they are often very connected to the surface area or the perimeter of an object rather than to the volume. So if you look at something like your small intestine, the surface area is the area which sort of this chemical reactions and digestions and these other things can take place. And so having a much higher surface area is very important, and having a higher volume isn't really correlated to very much. And so these are the things that are quite critical. And so we want to be able to analyze this. In material science, you see that this is very important for things like coarsening or rheological behavior. So like when foams are flowing, you're looking at these interfaces and surfaces. And then finally for like the chemistry processes. So if you're looking at like a catalyst action or certain types of diffusion, then the surface area is very important because that's where this is actually taking place. So when we look at surface area and perimeter, as we kind of saw a little bit before, the dilation and erosion is quite strongly connected to this. And so before, when we have sort of an object, when you perform an erosion operation or a dilation operation, you add or remove a layer of pixels at the boundary. And so this boundary is then the surface or the perimeter of a normal object. And so if we take a sort of very simple example with a circle, and we go from radius, so 0.5 to 5, and we measure the perimeter, and we measure how many pixels we add with a single morphological dilation or erosion, we have a pretty accurate estimate already for calculating surface area or perimeter. And so this is kind of an easy way to get into it because we've already covered erosion and dilation. And so applying this to an image is very simple, and it doesn't require much computation or very complicated algorithms that we need to implement. It's very much the same as what we've done before. So once we've done this, so this is kind of for a very simple object where we're just looking at perimeter. For 3D objects, then you can use this approach as well, but it's often not particularly accurate and not very useful for some of the other things that you want to do. And so here, the goal was just to calculate the perimeter or to calculate the surface area. And so doing one erosion or one dilation worked very well. But when we actually want to start doing something with this, that's then typically not sufficient. And so this whole area of taking a volume and turning it into a surface is called meshing. And so if you look at a large number of different tools, they will kind of refer to meshes or surfaces rather than volumes. and they're based fundamentally on the same data, but you have to make kind of a transformation to it. And the reason why meshes are so important is that if we look at sort of deformation as one very simple example, that if you have an image of an object before and an image for an object afterwards, and you look at all of the pixels, it's quite obvious for us that this object was squashed, but it's very difficult to quantify on a structure like this how difficult it is to turn a pixel off or to turn another pixel on. You know, we can find the average image, we can take the difference of these two images, we can do all kinds of filtering with these images, but it tells us very, very little about what's actually going on. And when we look at sort of a mesh, we have all of these different points here, and we have sort of the lengths or the edges connecting them together, and so that's what makes up this mesh. And when we move points on a mesh, we now have a very well-defined rule for what happens, so what the physical response is to trying to move a point on a mesh. And so this is where Hooke's model comes in as a very simple first approximation that basically you have your length at rest or when no other forces are being applied, and that if you try to pull this or stretch this from that original length, you then get a force in sort of the opposite direction. And so what this enables us to do is to kind of take something that was in a pixel world before and do physical analysis on it. And so this is the entire basis for things like finite element modeling and lots of other kind of simulation-based approaches, is that you now have information about what that force is. And so when you have a before and an after, you can actually calculate, what is the strain on this object afterwards, whereas with these images you couldn't do anything. You could take the difference and show that they're not the same, but other than that, you have no model for showing what that physically or mechanically means, and here you now have one. And so that's one of the things that makes these meshes so important. So, since we use kind of voxels in the images, we can use the voxels themselves as an approximation for this structure. And so, if each exposed face of a voxel belongs to the surface, you can then create a mesh from this. And so this would be basically the same as what we showed before with the perimeter and doing a dilation operation. But it's not a very accurate mesh. And so, your surface is by definition now always going to be square at all of the faces, because your voxels have square faces, and so your surface can only have square faces if you're using voxels to represent that. And so that's why a number of other methods come in. And in some of the exercises, you'll see that we've already used some of these methods before as sort of a visualization aid. But you can learn more about how they actually work by sort of looking into these image-based meshing methods and trying to understand the methods like sort of marching cubes better. And so we won't really cover this in detail because it's a fairly complicated approach, but it can be very useful for a lot of the things that you're doing. Usually when you do other analyses after this, it's quite sensitive to how you've done this originally. And so when you take an image and you perform marching cubes to get your surface, and then perform some kind of finite element simulation, you'll find that if you change one parameter on your marching cubes, your simulation changes completely. And so you really have to understand both this method and what you're trying to do with the data afterwards in order to do this accurately. But basically, the marching cubes approach tries to kind of fit in shapes by looking at the pixel values. And so rather than just saying every border is a voxel, you say every border is sort of based on its neighborhood. This is the shape that fits in there best. And so if you go to Wikipedia, as is linked in here, you can then see kind of what that means. And so basically you have these 15 different configurations, and it goes through each pixel in your image and tries to figure out which of these 15 is the best approximation for the region you're looking at right now. And what makes this particularly useful is that it's implemented in such a way that it can run very, very quickly on billions of pixels. Because if it took 10 seconds per pixel, you'd never be able to do any real image with it. But because it's been implemented well, you can then use it. And so if you go to the exercises that we had, I think after the third lecture, or the exercises for today, or any of the exercises that we've had before. Teeth fossil viewing. And so this is when we were looking at a fossil example. We kind of did some filtering, some segmentation, all of the stuff that we've covered so far. And then down here, we have marching cubes, which is part of this SciPy and scikit-learn toolbox, where you basically give it a volume and a threshold, and then it gives you the surface, so the vertices and the faces that you're trying to show later. And so here we just used it to make a nice 3D representation of kind of the fossil that we were looking at, but it can be used. You can then take this list of vertices and faces and operate on it directly. And so if you tried to calculate surface area, you could use that very easily to calculate the surface area of an object, or use that as a mesh for another tool. And here we didn't take any extra parameters, but if you look into this method more carefully, you'll find all the different parameters that you can give it and how you can tweak how this final volume looks. So yeah, here we then inverted the sample, and then we focused just on the air inside of this fossil. And that was kind of the rendering you had. But if your original image is kind of noisy and messy, then this is going to be even noisier and messier. And so the most important steps here are processing the image correctly, and then this will turn out fairly reasonably. But if it doesn't look good here, it's very unlikely to look good when you make this surface representation. We then did the same thing for all of the foams and the bubbles. So you have this measure marching cubes where you then show, as we have in the exercises today. Then marching tetrahedra is an even more complicated algorithm where you then have even more shapes that you can try to fit in. Depending on what you're trying to do, that might work better. Of course, with all of these methods, if you're trying to pick a face like this just based on its neighborhood, then little bits of noise and artifacts in your image will cause this to be massively wrong. And so the more complicated these methods get doesn't necessarily lead you to better results, because it becomes more and more sensitive to small changes and small fluctuations. Cool. So if we look at kind of what the focus is for today, so we have sort of this simple analysis that we've done, where you have a single cell. You came up with a bounding box last week. We now have an elliptical model for it. And it does a reasonable job. Certainly the ellipse is much better than the initial bounding box was at fitting this object. But we have some problems. And so if we look originally, we assume an entity consists of connected pixels. And so in many cases, that works well. But here, if we have 3 different entities, then we've linked them all together. And so possibly that's not what we want to have done. We want to have some way of saying that certainly if they're not connected, they're different objects, but even if they are connected, they still could be different objects. And so what sort of tools do we have for that? And then we assume that the objects are well modeled by ellipses, or a single ellipse. And so this is also in many cases wrong, because there's lots of objects that really are not a very good fit for a single ellipse. And so while that gives us a very easy way to define things like orientation and anisotropy, it might not be the perfect approach. And so, how can we try to divide this object into more than one shape? Or if we assume this is all one object, it would be useful for us to know kind of where the bottlenecks are inside it, or where the really narrow passages are, so that we could start to, look at, maybe this is a cell dividing and we wanted to identify all of the subcells as different objects or something like that. And then how can we come up with more specific metrics which we can characterize our shapes beyond just this elliptical model that applies to kind of every shape? And so that's what we'll start on today. And so today we have sort of this analysis of complex objects, and so how do we go beyond sort of the very simple analysis we've done to something more advanced? So the outline and everything is the same as before. There's a few new topics on here. Curvature is something we'll talk about a little bit, but we won't have that much. I mean, there are some exercises which cover this before, and there are some use cases which this is very interesting with, but there won't be much in terms of the actual lecture material that goes into that. So if you're interested in that, there's some links here, and then there's some exercises from last year that could be useful for you. So we've kind of already done this previously on. So we kind of have what's the motivation, which we talked about a little bit last time. Then distance maps, which are some of the most important tools that we'll cover in this class. Skeletons and things like tortuosity. And then thickness maps, which is kind of one step away from a distance map. And then watershed segmentation, connected objects, curvature, and characteristic shapes, and some of those ideas. So we kind of have these motivations as all of these questions which we couldn't answer particularly well with the single shape analysis. And so we have a number of different areas where these are relevant, and so this goes from topology to looking at distances and measuring diffusion and sort of other aspects like that. And so these are the questions that we are trying to answer with the tools that we work on today. So we have this exact slide from last time with this cell. And so what we'll start off with is this idea of a distance map. And so if we take sort of a small object region, we have pixels that are labeled as background and pixels that are labeled as foreground. And so one of the useful transformations we can make is called a distance map. And so what we do here is we say that each point in the map is the distance that that point is from a given feature of interest. And so in the standard definition of the distance map, it's each point in the foreground, how far it is from the closest point in the background. And so if our image starts off as a collection of foreground and background, we can define the distance map operator that transforms that image into a distance map. And so the distance is the minimum distance here for all points in the background. And so we use a Euclidean distance, so just a very simple square mean of a sum of squares there. But there are lots of other metrics which you can use for distance. And so based on how your object looks and how processes in your object work, other metrics might be better. This is then also important, as we talked about last time, with sort of medical imaging devices, that you very often acquire anisotropic image data. And if you acquire anisotropic image data, so the slice thickness is much larger than the resolution in X and Y, then you can't use a simple Euclidean distance metric. You need to use some different metric because you'll be measuring things in the Z-axis the same way you're measuring them in X and Y. And so you need to somehow account for the fact that things in Z are much further apart than things in X and Y. But for most standard images, you can use the Euclidean distance metric and it should work fine. And so this is sort of what that distance map looks like, so that we take the image we had here, which was labeled as background to foreground, and we now have the distance from the background or distance from the object, whatever you want to call it. And each one of these pixels has a value between kind of 0 and 4, because 4 is the furthest distance we have right there. And it shows what that looks like for these objects. And so of everything in the background has the same value. It's all 0 because it's all immediately connected to a background pixel, and the things in the foreground then have positive values. You can use a different distance metric. So here we try like the Manhattan distance metric, which looks at city blocks. So how many sort of up or down movements do you have to make to get to a single pixel? And then you get a distance map like that. So you see that, you have a bunch of 4s here instead of a 4 and a 3.6 and the sort of other associated values. And so for certain techniques, this might be better. This is also much more efficient to compute, so it takes much less time. And so if we were to look at it step by step, if we start off with sort of a mask image, how does this then transform to the distance map? And this is what it looks like. And so we start off with an original image like this, and as you do each iteration, it gets more and more of the pixels that are further away from the surface. And so if you kind of think about algorithmically how something like this is implemented, that it is kind of like progressive erosion operations or dilation operations, where you then see how many operations it took to get to that certain place. And so if you label everything that's one erosion array is a 1, and then the next step is kind of 1.4, and then the next one is 2, and you kind of get maps like this. And so that's what that looks like. Again, this is when sort of the major precaution is that you need to have, if you want distance maps to be physically meaningful, then you need to make sure that they are well sort of matched to your image acquisition type. And so as we mentioned before, if you have isotropic imaging, then using a Euclidean distance metric works perfectly fine. But if you have anisotropic imaging, it won't work well, and the distances you get on your distance map aren't particularly meaningful. And so ideally, as part of filtering, you would kind of resample and make your image isotropic, because then you can use a distance map exactly as it is. But if that's not possible, you'll probably have to do some sort of custom algorithm or adjustments to make the distance metric calculated correctly. And so that's obviously much more work than the one above. So if you take the approach above, you can use algorithms exactly as they are. And if you use the approach below, you'll have to make your own distance metric in order to calculate it. And so now we can take an object like this, which is sort of a slightly zoomed out version of what we were looking at before, where we have sort of 4 circles. And we can create 2 different distance maps from this. And so we can either say the circles are the foreground and everything else is the background, or we can say the circles are the background and everything else is the foreground. And so that gives us 2 different distance maps that we can interpret in sort of different ways. And so if we look at this map, each pixel here shows us how far a point is inside the circle from the boundary. And if we look at the map here, it's kind of the spacing between the circles. And so if you were looking at kind of a biology problem here, and these were blood vessels, and you wanted to know for each, which of the regions that are supplied very well by blood or nutrients, and which are the regions that are quite poorly supplied, that you could use that background distance map, and you could see that sort of all of these green areas are very well supplied, but these sort of blue and purple areas are very far away from the closest vessel. And so now you have kind of a map for quantifying an idea like that, so that if you were to look at this image in more depth and try to analyze, for example, what the density was, or how likely it is to have a fracture there, or for a disease to grow, or something along those lines, you have a very easy way for calculating that, rather than trying to come up with some approach where you kind of measure the distance and you have to find all the different objects and compare it to the center of that object or all the pixels in that object, which gets very, very tedious and time consuming. And this is just for a very small image, but you can imagine that when you have a larger image with millions of pixels or billions of pixels and thousands of objects, that calculating the distance to the nearest object can be very, very time consuming. And so if you can use some map like this saves you a lot of time because you just make it once and then you can reuse it for a lot of different tasks. So one of the things that's very useful about the distance map, particularly when we compare it to the approaches we covered last week, is how insensitive it is to small changes in connectivity. And so this connected component labeling, which was kind of our primary tool last week, we saw that if there's just one pixel difference, you go from having, two components to one component, which makes your shape analysis very different, which makes your volume estimate, your surface area, all of this anisotropy, all of those values change massively when one single pixel flips. And when we look at a distance map of sort of these four small circles, we have them sort of in the standard configuration where they're all separated by a little bit, and then in the configuration here where they're all touching. And so they're just slightly closer together. These images are almost exactly the same. And we see that when we take the distance map and look at the distribution of how many pixels are how far away from the surface, that looks very similar. So there is a slight shift here, but it's almost the same. And if we were to take the volume measurements, obviously the total volume here is conserved, but the volume per object gets multiplied by 4 just because these pixels are touching. And you go from having perfect circular objects to sort of a blob object that has a hole in the middle that's much less uniform and a much less good fit for something like an ellipse. And so this is one of the things that makes distance maps very useful for us, is they allow you to kind of quantify an object and not be incredibly sensitive to small changes in threshold or position or other parameters like that. And so that's one of the reasons why this tool is so important, because single pixels tend not to make that much of a difference. Obviously, in the other extreme, here we had pixels at the boundary. If you were to add a pixel in the middle of this object, this distance map would then change substantially. So it's very sensitive to pixels in the middle, but those are fairly easy to remove. But in terms of the boundary, it's fairly insensitive. And so if we go back to the cell that we had last time, we can do a distance map on it, where we now have the distance from the surface of the cell for all of these pixels. And so now what we can see is that we have sort of a map for how far each point is away from the surface, and this is potentially a much better representation for our cell for doing other kinds of tasks. And so if we wanted to divide the cell into 3 different pieces, how could we do that? Any ideas? Yeah, so we could simply say any value that's further than 2.5 or so away from the edge is a different object. And so that would then break our object into 3 different parts, because these parts are very low and those parts are very high. And so that gives us a very simple way of breaking that thing up, where if we just had sort of a threshold image, we would just have black and white values and there would be no clear point that we could use to kind of divide our object into pieces. And so yeah, that would work quite well for that. and that would allow us to sort of split it up. We can then have the background image from a cell, which would give us information. If this cell is supposed to be communicating with other cells, you can now easily measure how far it is away from other cells and what other cells might be touching it. Where if we just had the center of volume or the center of mass, that would not give us a very accurate representation of that, because this point is very far from the center of mass, but it's very close to the actual cell. And so having a map like this can be useful in a number of different circumstances. So now, kind of moving beyond that distance map itself, one of the very useful things that we can do with a distance map is making a skeletonization. And so some structures like cellular materials, or in the exercises today, one of the examples is looking at aerial images with streets. But there's a large number of things that this applies to fairly well. And so we have an object that, let's say, looks like this. And for us, as sort of visual observers, there is a very clear topology to this object. So you have kind of a number of objects here, and they're all kind of linked together by these kind of channels that go through there. And so here, that's the background, and this is the foreground. And so that's easy for us to see, but if we think about the tools that we made last time, that doesn't tell us anything about this object. Anisotropy isn't really interesting. The area or volume isn't particularly interesting. We're interested in kind of what are these little subpieces and how are they connected together. And where might be the bottlenecks. If you were a chemist, you might be interested in sort of, if I put in a flow here, where is it likely to come out? And so that would then require some kind of graph or network representation of what's going on here. And so, yeah, these are kind of our tasks, where the first is kind of which structures are connected together, how well are they connected, and then how can we express this network? So we don't want to have 10,000 or a million pixels that we need to somehow analyze. We want to have a very simple representation of the network. And then how can we start to quantify more interesting variables? And so for the chemist or potentially neuroscientists, things like tortuosity, so how much kind of curving around does a structure do before it gets to its endpoint, is very important, because the more curving around it does, the slower a flow will be through that. And then for biologists or like cardiac specialists, knowing about the branching can be quite important. So if you're looking at sort of heart tissue, knowing about how much those tissues branch and what they're connected to is quite important. That particularly if you're looking at disease states where, tumors might be forming, that they have a lot of angiogenesis, and so they have tissues where there's a lot of branching going on. And so if you can somehow quantify how often are these little vessels splitting into new vessels, that's quite important. And none of those things are things that we could have done before. And so these are the challenges that kind of make skeletonization useful for us. And so one of the first things that we can do is we can sort of use our distance map like we had before. And so we can take our image and sort of transform it with this distance map, and then look at where the values from the surface are. And so we can already see by looking at this that we have a much better representation of our image by just making this map out of it. And so what's helpful is that, if you look at that image, you can now say that most of these local maxima correspond to sort of this backbone or this network that we were interested in before. And so we have here kind of these hilltops or whatever, if you imagine that as a topology map, where that is the structures that we're actually interested in. And so if we could somehow trace a line along this, we would now have a perfect map for where the skeleton of this object goes. And so we can now take this and apply some of the tools that we've covered before where, if you remember from the second lecture, we had these different kernels that we could apply to images. And so one of them was like this Laplacian, which is kind of a derivative operator. And so we can then use that to find where the high gradients are in the image to help us try to find these local maximum values. And so we can apply that to the image, and we end up with this sort of output where we can see sort of most of these values are flat, and then we have these lines that represent where the backbone of the structure was. So we can now try to create a skeleton from this, where we sort of have two requirements that we try to combine together. The first one is you don't really want anything at the surface, because we see here all the surface pixels have a very, very high second derivative, and so that isn't very interesting for us. The border of the object we identify in other ways. That's not what we're trying to do with this tool. And so as soon as you get away from that, then you can focus just on the values where you do have the high second derivative, or this local maximum. And so you can come up with rules like that for making a sort of feature vector and then applying a threshold now to this image to segment out exactly what we're interested in. And so, you can try different thresholds where, if you have a minimum slope of 1 and a minimum distance of 2, you end up with the blue points, which are the points that sort of have this high enough Laplacian value, and the pink points, which are far enough away from the surface to be considered sort of eligible for being part of your structure. If you then take sort of a minimum slope of 0 and a minimum distance of 1, you now have kind of all of these tiny little substructures, and you kind of have to balance which things you're most concerned with. And this would be then very determined by your resolution, the size of the structures that you're looking at, and the size of the branches and how many branches you expect. Because if we go back to this original image, it's quite obvious for us where that backbone is. But for an algorithm, it's unclear, or it's not initially very well defined. Is the backbone this, or is the backbone sort of going along to every place where this isn't perfectly round? Or are there sort of other features that might be useful for that? And so, particularly with very thick structures, it can be very complicated to get just the features that you want. And so you often end up with a lot of these little hairy things coming out, which is why pruning becomes important. And so if we take that structure from last time, we end up with a skeleton that has many more points in it than we're actually interested in, and then we can prune that structure. And so what that is tools that allow us to kind of go through and remove branches that don't make sense or that are too close to other branches. And so you kind of have a model where you say, the minimum branch length that we would expect is this, the minimum branch width is that, and then you can go through and get rid of a lot of those smaller sub-branches so that you have a clean representation of the object that you're trying to look at. But there is no magic formula for always getting to the right skeleton result. Cool. So now once we have our skeleton, and ideally our skeleton is then 1 pixel thick in all of the areas it is, we can now start to do more useful things with it. And so for branching, or for this topology and network approach, the first thing we want to do with a skeleton is to start classifying the junctions that we see inside of it. And so if we take an image like this, we can quite easily classify the points into different things by using the neighborhood values of each pixel. And so we have here sort of structures that are going through. And if we look at a point that's sort of in the middle of this segment here, which is what terminology we're using, if it is touching 2 other points, then that means that it's a segment. And if it's touching 3 other points, that means that it's a junction. And if it's touching 4, then it's also a junction. And if it's here and it's only touching 1 point, then it's sort of the end or the terminus of one of these branches. And so that we can go through our image and apply these different algorithms in order to see where are the junctions and where are the terminals in our image. And so with these 2 images, what we see very clearly is that this representation, because it's too thick, gives us bad values. And so we see that that's correct, that we have sort of this green classified as segment here, but all of this is classified as junction because it's 2 pixels wide. And so that's not something that we want to have. Similarly, on this side, we have a little bit of green here, but we have a lot of junction pixels there, which are not useful for us. The terminals seem to be okay, but we even have some points that are classified as terminals that we wouldn't necessarily have identified as terminals. You can then use a box neighborhood, and a box neighborhood is then even worse in terms of how many points it finds, but you end up with kind of a similar map at the end. And so we now have a bunch of metrics we can start to assess with our skeleton. These, of course, were not great examples, but they give you an idea of the things that we're trying to do, where you can start to count the number of junctions, segments, and terminals in order to see what the relevant structures might be and how your structure compares to another structure, and counting things like branching and growth and changes and everything. And so once we have these classifications, it's very easy for us to analyze a number of different features in that image because we've classified our object in these groups. So I think we'll take our break here, unless there's any questions on that so far. No? Okay, so we'll take a quick break here, and then we'll resume in 10 minutes or so.